Copepods, small crustaceans found in marine and fresh waters, are important for aquatic ecosystems. They consume phytoplankton and serve as food for larger organisms, including fish. Copepods could hold clues to improve our understanding genome structure and evolution, providing novel genome editing tools. Some species have the unique ability to remove DNA sections from their genome during embryogenesis. Copepods have a multi-stage life cycle, including embryo, larva, juvenile, and adult. Somatic cells make up the body, while germline cells produce gametes. Females carry embryos in egg sacs until they hatch into larvae. DNA loss, known as programmed DNA elimination, occurs during an early embryonic cleavage division. DNA elimination occurs only in cells destined for the soma. Copepods eliminate DNA through loop formations. Fragments form granules resembling autophagosomes. Typically, 50 to 90% of DNA is excised. Somatic genome sizes are reduced in daughter cells. The germline preserves the original amount of DNA to reproduce programmed DNA elimination in the next generation. The eliminated DNA ends up in the cytoplasm. DNA elimination occurs synchronously. Excision varies in timing, location, and amount among species. The majority of the dark matter DNA is comprised of potentially deleterious transposable elements, which can move around the genome. Most organisms defend themselves against these elements using a form of gene silencing. Copepods go a step further. They excise these elements. The identities of excised DNA point to hypotheses of adaptive functions. Excising transposable elements protects the soma from potentially deleterious effects. They may promote their own excision, perhaps to avoid harming the soma while they reside silently in the germline. Almost 1,000 genes are excised. Most are linked to gametogenesis, suggesting a form of gene silencing or gene regulation. DNA elimination also modulates somatic genome size. Smaller genomes have faster cell division rates, increasing chances copepods will reproduce before a fish eats them. Excised DNA may provide metabolites for differentiation and replication and adaptation in low-nutrient environments. These hypotheses are not mutually exclusive. Programmed DNA elimination of the kind that involves chromosomal fragmentation is observed in ciliates, nematodes, Japanese hagfish, and sea lamprey. Comparative genome sequencing may reveal if this trait has independently evolved. Mesocyclops edax is a well-studied model for programmed DNA elimination. Non-coding repetitive sequences, known as the genome's dark matter, have unclear functions and make up most of the genome. Genome size varies among eukaryotes due to these sequences. Genome sequencing of germline and somatic genomes is providing knowledge to identify DNA break sites and the molecular mechanisms of DNA excision. The foundational histological and genomic groundwork has been laid. We are poised to move forward and understand the evolution of programmed DNA elimination in nature, as well as how it might be harnessed in the laboratory to improve health and agriculture. DNA elimination provides some of the raw material for genome evolution, perhaps even speciation. One might think DNA loss is deleterious, but instead it may lead to genome stability. Understanding the mechanism of DNA loss would initiate a new era of genetic engineering. Like CRISPR, selectively programmable genome editing tools have the potential to cure genetic diseases, breed more resistant crops, and usher in advances in biotechnology which have yet to be imagined. Studies of transposable elements and programmed DNA elimination are frontiers that are inextricably intertwined. We are poised to explore them.